right, guys. So, with the cell membrane, these are the things that stood out to me as I was reading it. Understanding, obviously, the hydrophilic heads and the hydrophobic tails. Knowing about integral proteins that are transmembrane. Knowing about peripheral proteins, which are on the outside of the membrane. Knowing that cholesterol is the temperature buffer there. I thought it was interesting that the membrane's like an olive oil consistency. I wrote that down just because I thought that was interesting. And then also one thing that I thought was important is the connection to the cytoskeleton. One of the things I want you guys looking for as we went through these presentations, I hope you noticed, they talked about this at the Golgi, and then they talked about this other thing at the cytoskeleton that went with that, and then they talked about this at the membrane, which went with the cytoskeleton, right? Like, so there's connections with all of these organelles, and so hopefully you caught on to some of that as you went from presentation to presentation. And so one of the things that I thought was really important at the cell membrane is they talked about its connection to the cytoskeleton. And the cytoskeleton is going to talk about how it shapes the membrane, okay? So one thing I wrote, and I'm not writing on like the fill-in notes. These were just things I jotted down as I listened. But one thing that I wrote down that I thought was an important cross connection is that the inside of the membrane connects to the cytoskeleton, the outside of the membrane connects to the extracellular matrix, okay, which is what connects cells to cells, especially in animals. So those are the big things I wrote down for that one. Okay, let's talk nucleus. That's the next one on my list. Nucleus, you wrote your own notes, I think. Okay, on the nucleus, I wrote down, and so this would have been Sydney or Ashland presenting to you. I wrote down about the double membrane. I mean, we had talked about that too, but that's obviously something you need to have locked in. As the nucleus is not on a double membrane. And that outer membrane is continuous with the ER. The ER is going to talk about that. So you could write that on either note you have. The envelope versus the lamina. Like the lamina is kind of that net-like array of protein filaments. I wrote down about how most cells are going to have chromatin, right? But when the cell's dividing, we see chromosomes. And something you may need to add, they talked about in the nucleolus how our RNA is combined with proteins. When our RNA is combined with proteins, what have we made? Ribosome parts. Okay, the two halves of the ribosome. So they talked about our RNA combining with proteins inside the nucleolus. Well, that's making the two halves of the ribosome that you learned about at the ribosome station. Okay? That's the main stuff I've got written down there. All right. Chloroplast is my next one. Dave, fill in notes. These chloroplast and mitochondria get a little tough because we're gonna spend way more time with these two as organelles go. And so there's a lot of information there, but what you really need to take away for this chapter is probably not as deep as what we learned. I have the structures written down for the chloroplast, so like thylakoid membrane, stroma, Okay, the grana, which is a stack of thylakoids. So kind of knowing, right, the basic function or the basic structure of a chloroplast. And then the other, knowing that obviously this is where photosynthesis occurs. We're converting light energy, or the plant's converting light energy into chemical energy in the form of glucose. And then the big thing from there is that you need to know is the endosymbiosis, which I think is at the end of their notes. So endosymbiosis is this idea that because chloroplasts and mitochondria, this is true of both chloroplasts and mitochondria, they have their own DNA, and that is super unique. They have their own DNA, they have their own ribosomes, and they can reproduce on their own inside of the cell. So it's not like they reproduce when the cell reproduces, they reproduce when they need to reproduce. Okay? What does that sound a whole lot like? A bacteria. Okay? They reproduce the same way bacteria reproduce. They have the same shape DNA as bacteria. They have the same structure as bacteria. And so the endosymbiosis theory is that at some point, cells engulfed chloroplasts, and instead of breaking it down, they were so productive for the cell that they just began reproducing together. Okay? Over time, all plants had this enclosed chloroplast. And mitochondria, the same thing, that they were cells living by themselves, like bacterial cells, okay, engulfed by other cells, and were so useful that they reproduce with those cells now. Okay? And so that's a big one that you need to know from that presentation. Any questions about that? Because that is like a new concept. All right, next one I have is mitochondria. You have fill-in notes for this too. This was Lauren and Allie. Chloroplast is Lauren and Maddie. Okay. Again, have their own DNA and ribosomes, right? These are the only two organelles that fall into this group of having their own stuff. They are producing or doing cellular respiration. And in fact, 90% of energy is made in the mitochondria. I had that written down. One big thing, they talked about the cristae, which is the folding of the mitochondria, structure-wise. And this is a big one. The reason it's folded, and they said it, is because there's more surface area to make more ATP, right? The whole idea of having this folded cristae inside the mitochondria is so there's more surface area to make more ATP. Structure and function go together in all of these organelles. Okay? And that's really the biggies. Okay? Those are the biggies for that one. Any questions about that? All right, next one I have is smoothie art. You guys wrote notes for this one. All right, smoothie art makes lipids, and the most important part about making lipids is it's making cell membranes. That's really big. So all these things that have cell membranes, the cell that has a cell membrane, all these membrane parts, these phospholipids, are being made in the smoothie eye. So that's a big deal. The other big one, I mean, it metabolizes things like carbohydrates, but it also metabolizes drugs, so like detoxifies drugs. And we've known that. Like, I think that's what should have come to your mind for the smoothie eye. And one important thing there is that the more it needs to work, the more it reproduces itself, right? And we've talked about that. So it's like you're a habitual drug user, or like even like a certain prescription drug sometimes, you will have huge amounts of smoothie eye. Like your smoothie eye, what's the word for that? It starts with a P? Proliferate. your smoothie R will proliferate. That means it'll just keep reproducing to keep meeting the need to detoxify. And the other thing that they pointed out that was important is it makes calcium for muscle contraction, so your muscles will have a lot of smoothie R too, because it's doing muscle contraction. So those are the biggies that I had. Next one I have is cell wall. This is a big one with a lot of information. All right. Cell walls are in plant cells, some prokaryotes. I thought one really important thing, like their diagram showed it really well. It's much bigger than the cell membrane, right? Like it's much thicker. Really changes the cell. It hardens, like when it stops growing, it hardens, which is what gives plants their ability to fight gravity. Plasmodesmata are these joiners. You needed to know about the plasmodesmata. It's kind of like a key feature of the cell wall, and they did a good job of explaining that. But it's what joins plant cells together and really makes it multicellular, right? Like it's really what's connecting everything. I wrote down like primary lamella and the secondary wall. I don't know. I mean, that's a lot of detail, but we won't spend much more time in the wall. The lamella is what sticks the cells together. Like it's actually, sticky. I think those are the biggies. Water can move through the cell wall. Macromolecules can move. That's probably an important thing. All right, my next one is cytoskeleton. You have fill-in notes for this. So you kind of, I think you wrote, there's a paper with a few, but you wrote extra things probably. Cytoskeleton's super cool. Like this is the track inside the cell that everything moves on. So one key thing is like motor proteins move things along this track that like goes all over the cell, different tracks. And so that's a really big part of it. And really for this one you need to know microtubules, intermediate filaments, and microfilaments. Those are the three sizes. One cool thing, like it can break and reform. Like it's what makes cells different shapes, animal cells different shapes. Is its ability to even like at the spur of the moment break down and rebuild somewhere else. It makes me think of like Big Hero 6 when like those little bots, like can go down and then rebuild and like move all over. That's what I think. That's what I'm thinking of in my head when I'm thinking about the cytoskeleton. And stuff moves along them. That's what in my head. That's what it's like. And it's pretty accurate actually. Anchors, organelles, like this is what organelles are on. Like they don't have their own movement, and so if an organelle needs to move, it's moving along this. Any questions about that? All right, next I have the Rephiar. This would have been Emily or Eric. This, I think you wrote notes. Found in eukaryotic cells, right? cisternae are the folds in it. The ribosomes are attached by microtubules, like so part of the cytoskeleton is working to attach, and they can move around a little bit. And the big deal here, obviously, is they're making proteins to exit the cell. So it's going to go from here to the Golgi. Ruffyard. What? Cisterna. Like Cisternae. And so making proteins that are going to go onto the Golgi that are going to exit the cell. Okay. Next one I have are ribosomes. You took notes on this. By hand, I think. All right, ribosomes have two parts. They were made in the nucleolus. They are building polypeptide chains, bound or free. I mean, this goes with what we just said in the Ruffyard. So much of this, guys, goes together. Hopefully you're like sensing that, seeing that, right? So the bound are bound to the nucleus in the Ruffyard, and those proteins are going where? Out of the cell, right? The free ribosomes are making proteins that are going to stay in the cell. Not truly a organelle because it's made of RNA. I mean, kind of unique in that way. Not membrane bound. All right, lysosome, central vacuole, peroxisome. Anasazi and Bitland. All right, lysosome, big thing, full of hydrolytic enzymes, so it fuses with like food vacuoles and breaks stuff down. Breaks down food, breaks down old organelles, kind of recycles molecules to the cell. Central vacuole, huge part of the plant cell. What gives the plant cell kind of its structure? It pushes against the cell wall, so it can be really full of water and other molecules. Little food vacuoles in animal cells. And then peroxisome. So our cells make hydrogen peroxide as a byproduct. It's toxic. Peroxisomes break down hydrogen peroxide. Yeah. And so the big thing about peroxisomes is they break down hydrogen peroxide, which is toxic to us. It's poisonous to us. And so they are basically like a lysosome that is specific for hydrogen peroxide. And last one, the Golgi. Gorgeous apple salad. Yeah. I have it written down. They were. All right. You need to know the cis side is where the vesicles are received from the ER. The trans side is the part that sends out the modified protein. They take from transport vesicles and they send out transport vesicles. What are transport vesicles made of? Oops. think about it for a minute. So where does it, and this isn't like, I just want you to think. So what happens on the reticle? The ribosomes do what? Make protein. And what happens to the protein that's made on the reticle? It goes out of it's going to go out of the cell eventually, right? So what does the cell put it in? A vesicle. And where does that vesicle go? To the Golgi. And then it like modifies it, right? And then where does it go? Out of the Golgi, right? Out of the transite just by itself, or in what? In another transport vesicle, which takes it where? Where? And where you got to go to get out of the cell? Through what? Cell membrane, right? And so you've got like all of that, guys, is part of the endomembrane system because all of it's membrane. All that membrane is interchangeable. We may go back to that out. Okay. The other thing, like one of the main things that are produced in the Golgi are glycoproteins. What's a glycoprotein? It's got like a sugar attached to it, right? And you heard about glycoproteins in the cell membrane presentation because they're used as like kind of identification on the cell or to bond to other cells. And so those glycoproteins are just a protein with a little carb attached to it that a lot of times is used as like an identification tag. Like, hey, we're liver cells. Let's all get together. Okay, make a liver. So all that goes together. And that all happens in the lumen, which is the middle part of the Golgi. Lumen is kind of a word, like lumen we heard a couple times. It kind of is used in more than one organelle. Okay. Questions you guys have for me? Okay, let's try to see if we can go do a very multi-part. Oh.